Right, welcome to this video on biology. This is the QCAA Queensland Curriculum Authority, Queensland Curriculum and Assessment Authority exam paper 2023. I'm doing these papers just to give you a bit of a hand with your revision maybe. The exams are coming up in November, I guess. And also to give me a little bit of practice at the curriculum because I haven't taught in Queensland since 2018. Things have changed a lot since then, and I may end up going back, so I don't know. Let's see how things go. But anyway, this is not about giving you the answers for this because you can easily get the answer sheet from the exam from the QCAA website. This is about thinking about how you answer questions. So let's see how we go. All right. Let's see if we can get through this. This is obviously a printout. I haven't got the original paper. So it says here, instruction, respond to these questions in the question and response book. So you don't put your answers here, but we're going to anyway. Right, let's have a look at question 1. The biological species concept defines species as a group of organisms. And then we have 4 choices. We have to pick one of these choices, okay? So, with a common set of alleles, well, that's not a species. That would be very, very close family members would have a common set of alleles, and even then, they're not likely to be the same set. If we have a clonal population of animals, say for example laboratory mice, they may all have a common set of alleles, but even then, it's not certain. Things can creep in, genetic differences can creep in. But anyway, that's not the answer. It's not a species. It's a very closely related group of organisms. Would be a descended from a common ancestor. Well, all species are descended from a common ancestor, of course, the last common ancestor, as it were. So that's not true. Occupying the same niche or adaptive zone. No, this would be, we're talking about maybe more than one species occupying that niche in competition with each other. That can interbreed to produce fertile offspring. So that's the answer we're looking for. They can interbreed to produce fertile offspring. This definition of a species doesn't necessarily cover everything, but we're gonna go for D. Doesn't cover everything like bacteria, things like that, because obviously things that do not breed sexually don't have to worry about this. So there you go. Okay, question 2. Let's see. Question 2, species classified as K strategists. Okay, what are the K strategists? The way to remember this is the K strategists are the ones that give care. Care. Remember care with a K, right? And for me, the other guys in this, they give care, right? Parental care. K strategists, parental care, okay? So they're the ones who look after their young, put a lot of energy into rearing offspring. The other ones are the R strategists, and I think of them as the fire and forget. So the fire and forget strategists are the R strategists. They're the ones that tend to lay eggs and then just swim off and leave them to their own devices. They don't put a lot of energy into each individual egg because they're not expecting very many of them to survive. Anyway, they're talking about the K strategists here. Species classified as the K strategists. So they often live in unstable habitats. No, you're better off being a fire and forget job there than R strategists. Exhibit an exponential rate of reproduction. No, that's again, it's an adaptation to an unstable habitat. Reach sexual maturity later than R strategists. Well, at least they've mentioned the R strategists here, so in case you get confused. And are first to colonize a new environment created by disturbance. So our answer here is these 3 things here, A, B, and D, are likely to be R strategists. The only one here that is likely to be true here is the K strategist. They grow more slowly, they reach sexual maturity more slowly, they're given more care when they're young. Therefore, they're bigger and they're older and they're stronger by the time they're breeding themselves. Okay, and our final question here on this page is, the table provides population data for a species of fairy wren. All right, population growth rate in 2022 was closest to, this is 2022, here's the data. Let's look at the table. So that's the year, population on 1st of January, that's the beginning of the year, so that's the population at the start. So start, write that in, just make it handy. So we've gained, that's a plus, we gained 7,600 individuals. We lost, that's a minus, 4,310 individuals. If they all survived, we'd be knee deep in fairy realms. The world would be a crazy place. Okay, a bunch of them have immigrated in, so that's a plus. They've come in, and immigration is out, so that's in and that's out, and that's a minus. Okay, so what we have to do is we have to work out what the change in the numbers added to the population were as a total for 2022. So I'm going to take my calculator. Hopefully we can see this. And I don't know if we can see it. We might not be able to see that screen very well. But anyway, what I'm going to do is I'm going to take 7600 minus 4310 plus 790 minus 24. Okay. We're going to add all those numbers up. And let's see what the calculator says about that. So on 760. 7600 minus 4310 plus 790. You can just do this all in one row in the calculators, no problem. Minus 24. That all equals 4056. That looks good. Can we just see the answer to this? 4056 looks like about 25% roughly. So I'm gambling on 27 being the correct answer here. So let's put the change over the start times 100 to get our growth rate. And that's going to be 4056 divided by 15,200 times 100, and that should come out, hopefully, that looks like it's going to come out roughly at 27%. So let's see, 4056 divided by 15200 equals 26.68 times 100 equals 26.68, 27%, and that's going to be our answer B. Okay. Let's move on to the next question, which is, okay, populations with reduced genetic diversity face an increased risk of extinction because, so reduced genetic diversity means they don't have many different alleles, basically, in their population. An increased risk of extinction because they have fewer chromosomes. No, they could have many chromosomes. That doesn't mean anything. So chromosomes don't indicate genetic diversity. They have difficulty finding mates for reproduction. Well, genetic diversity doesn't really have any effect on whether you find mates or not. That's more to do with your population. Are less likely to adapt to changing environments. All right, well, this looks like a good answer to me. Less likely to adapt to changing environments. And contain a larger proportion of heterozygous individuals. Well, that's not going to be true because they have low genetic diversity, so they tend to have more homozygous individuals. So this is more likely to be true. They're less likely to adapt to changing environments because the genetic diversity is low. Therefore, if the environment changes to the point where they're stressed out and they need a bit of variation, that's not going to happen, is it? Okay, which statement is true for DNA replication? DNA replication. Okay, so what's happening with DNA replication? DNA is making copies of itself. That's what DNA replication means. So remember, make sure you understand what these terms mean. And genetic diversity up here as well. Genetic diversity. You need to understand these terms in order to be able to understand how to answer the question. So a lot of the question is just asking you, what does genetic diversity mean? This is basically asking you, what do you know about? You need to understand what DNA replication is before you start. So vocabulary is so important. I've got a video on learning vocabulary. Go and watch it. Adenine pairs with guanine. That's not correct because adenine pairs with thymine and guanine pairs with cytosine. So that's not correct. The process occurs during metaphase 1. No, it's all over before metaphase 1. DNA replication happens before even the nuclear membrane breaks down quite often. Well, it will happen before the nuclear membrane breaks down, or else it's not going to work. DNA polymerase unwinds the double helix. No, it doesn't. New strands are synthesized in the 5' to 3' direction. That is true. Okay, that's good. That's basically the one that's true. So you've got your full strand coming in one direction, and then the Okazaki fragments build up in the other direction, 5' to 3' both ways, both sides, both strands. Okay. Oh, we can't do this one. So let's see, what was the answer in case anybody really cares? We can't see the graph, so we can't really interpret it. Graph, data, evolutionary, we can't really interpret it. What do they say in the answer sheet? They say question 6 is answer A. Can't do that one. Let's move on. All right, question 7. Distribution of phenotypes before and after selection pressure acted on a population are shown. Which graph shows disruptive selection? Again, disruptive selection. This is forcing phenotypes apart, where you might have a couple of different phenotypes in the population. What happens is that the genotype pattern is switched to you get a bimodal distribution basically of genotypes afterwards. Okay, so here we go. Let's have a look at the graph. The graph is before selection pressure, and then 25 generations later. So what we're looking for is a smooth bell-shaped curve, normal distribution before selection pressure, and then 25 generations later we're looking for a bimodal distribution. Okay, now there's a couple here that look a bit bimodal because you've got two distributions where the mode has changed, but that's not, we haven't switched it to a bimodal distribution of phenotypes. So here we have our normal bell shape curve. That would be what we're looking for at the start. And then, so what has happened here is the phenotype is narrowed. So the niche is probably, the realized niche has been reduced for this species. Here what's happened is that a phenotype has been selected. So there's been some environmental change where the phenotype has moved from this mode to this mode, and a whole bunch of phenotypes here have gone extinct. Here, same thing's happened in reverse. So basically you've gone from, so that was before there, it's gone that way. This one is going that way. So I got that mixed up. And here what we have is our original bell curve and our bimodal distribution. So what's happened here is that the population has been split into two predominant phenotypes. And that's the result of disruptive selection. So I'm going to go for D. Okay, good. Question 8, here we go. Is karyotype. We always seem to see a karyotype in these papers. It's interesting. So the diagram shows a human karyotype and a list of genetic conditions. There we go. There's a human karyotype. Let's see if we can see anything obvious in it. Well, we've got trisomy here. And what number is that? They normally put them in order, and they will be in order, I think. So 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16, 17, 18. I'm just going to drop an 18 down there. And let's see what they're giving us. Everything else looks normal. X, Y, it's a male. Okay, so we've got monosomy X. That's not true. There is only one X, but there's a Y chromosome as well. We have monosomy 5. That's not correct because the 5th chromosome has 2 copies. We have Edwards syndrome, which is trisomy 18. There we go. Trisomy 18, and Down syndrome would be trisomy 21, 19, 20, 21. Would have 3 here if it was Down syndrome and 2 here. So we're going to select Edwards syndrome. All right, right down the bottom of the page here is an ecological niche refers to, well, you just basically learn the definition of ecological niche. It's the role that an organism plays in an ecosystem. That role in space that an organism fills in an ecosystem, see it straight away. Group of organisms is not one of them. Competing for the same resource, no. All organisms occupying physical space close enough to interact with each other. Well, this is competition within space. This is competition within a niche or within a resource area. And largest population of a particular species that can be supported by an ecosystem, that's not true. That's the carrying capacity of an ecosystem. So we've got a number of definitions here. You just need to remember that that one refers to ecological niche, the role in space that an organism fills in an ecosystem. Let's move on. Done that. Okay, question 10. Graph shows the extinction rates over time. Extinction rates over time. Okay, there's extinction rate up here. Families per million years, right? And then we've got 500 million years, 450 million years, 400 million years, all the way to the present, because this is million years ago. So 0 million years ago is today. There we are. That's us. Okay. And what we're looking for here is how many times in the past 500 million years has the extinction rate exceeded 14 families per million years? Seems straightforward enough, that. There's 14 families per million years. If I took my ruler out, which I don't have handy, and drew a line straight across there, I'm going to see it's exceeded that number 3 times. Is that it? Seems to be the answer to the question A. 4 would be wrong. There don't seem to be any traps. Seems a nice straightforward question, that. Okay. And then question 11. Restriction enzymes. Again, you just need to know what they are. Yeah. So they join DNA into a single strand. That would be DNA ligase, I guess. But that's certainly not the restriction enzyme. Cut DNA at specific locations. That's the one we want. Add nucleotide to a growing DNA strand. That's DNA polymerase. Assist in the amplification of recombinant DNA. Not quite sure what would do that. What would do that? Some other form of... That would be assist in the amplification of recombinant. It's something to do with PCR, possibly. But anyway, we're going to take this one. Cut DNA at specific locations. Some of them are great names. Actually, my favorite was BAMH1. They're named after the bacteria that they come from. Echo R1 comes from E. coli, for example. Okay. Let's turn over and see what we got on the other side. Very good. Question 12. So looking fairly straightforward so far. The graph shows the results of a study on the species richness of gut microbes. The data represents the cumulative number of species observed as the number of samples increased. So here's our number of samples. We took lots of samples and counted the species of bugs in their gut microbes, okay? And it wasn't until we got to 12 that we got the lot, okay? So once we went past 12, we weren't getting any increase. I mean, we may have hit the law of diminishing returns here where you're just counting the same ones and there might be rare ones you're not counting. But in the end, you can't do any better than that. So with 12 samples, you're getting most of the species, right? Anyway, based on this data, or these data, it's plural data, yeah, these data, the minimum number of samples required to obtain valid species richness data is, I'm going to say 12, right? Minimum number, yeah, but we would get it with 16. Yes, but that's not the minimum number. And we would not get it with 6, and we certainly wouldn't get it with 2. So I'm going to go 12. Question 12 is C. Okay, which option best describes the conditions for parapatric speciation? Yes, this is a fairly controversial form of speciation, actually. This is speciation that occurs in a huge area where... populations within a species that are spread right across that whole area maybe don't move very much, and so they have to just reproduce with the ones next to them rather than go hunting for the best mate or whatever at the far side of the area. So for example, you've got a continent like America, you've got things like cockroaches and stuff. The cockroach in New York isn't going to pop over to Los Angeles to see if he can find a better cockroach mate. He's kind of stuck with the ones he's next to. So anyway, let's have a look. Geographical barriers limit gene flow between populations. No, that's not parapatric speciation. This is oh God, where's my knowledge of? I can't remember what these. That's. It's not sympatric is similar to parapatric. I can't remember the other term. Anyway, gene flow is interrupted in populations occupying the same habitat. That one might throw us. Gene flow is interrupted in populations occupying the same habitat. I don't think that's going to be the case. There would have to be something actually happening to the species, and that's not the case in parapatric. Parapatric is just, it's a theory that says you just mate with what's next to you, and that kind of tends to group phenotypes in certain places. Species occupy such a large, oh, here we are, large geographical area that mate selection is influenced by proximity. There we go, that's the one we want. And a small group of organisms becomes separated from their parental population by physical barriers. Allopatric, is it? Allopatric, that's allopatric, and then we got sympatric, is same. Anyway, this is the one we want. C, a species occupies such a large geographical area. That's the one you're looking for parapatric anyway. Okay, let's move on. Must check out my speciation notes and see how we go there. Okay, an energy pyramid for an aquatic ecosystem is shown, and the values are in kilojoules per mass per year, basically. So let's see. Kilojoules of body mass per year or something like that, is it? Is that, okay, so let's see. These are the numbers. This is the energy. It's in kilojoules anyway. And the producers have 21,600. The primary consumers have 3,400. So it's not to scale. That's what they've set up here, not to scale. Because that, if it was to scale, they wouldn't have room for their text. All right. Then the secondary consumers 410 and the tertiary consumers 20. So that would just be a tiny little line. Okay, the transfer efficiency between producers and primary consumers is closest to, well, make sure you get the producers. So producers is, let's call that X, and then mix that. And then primary consumers, I'd call that Y, just to make sure we don't get mixed up. Okay, so those are the two we're looking at. I mean, they could have picked any of these and you might get mixed up, so make sure you get them right. Okay, transfer efficiency, this is basically a change, percentage change. So again, we're going to put our efficiency is going to equal, it's going to equal the end, which say this is the end result over the start times 100. Usually you just put in the little number over the big number, but anyway, it's going to equal, so end is Y, 3400. Producers is 1600. Now what's this going to be? Looks not very big, but these aren't very big anyway, so let's do the calculation on 34 divided by 2.600 equals 15.7, 16. We're going to go for 16, D. Okay, 14 is D, yep. You probably can calculate some of these if you do this wrong, but you can't get it. I mean, if you put these numbers upside down, you're not going to get over 100, so it's not going to, you might get I don't know. Anyway, let's move on. Allele frequencies are most likely to stay constant in. Okay, here we go. Large populations with low levels of migration. Large populations are more likely to keep allele frequencies constant. So I think we're going to remove small populations from our thinking here, because small populations tend not to have constant allele frequencies. Okay, and low levels of migration will tend to keep allele frequencies low, because high levels of migration is going to bring alleles from outside into the population. So on the basis that we're talking about population size and levels of migration, large populations with low levels of migration is going to be the correct answer, isn't it? Right, that's the next question. Let's have a look. Okay, the diagram shows a section of DNA. There it is. Here's our section of DNA. We've got grey bits and white bits. The grey bits are the exons, the white bits are the introns. Again, you need to know exactly what the words mean. So the exon is the bit that is converted into messenger RNA to make a gene. The introns are the bits that are cut out during splicing. Okay, and then we can get alternative splicing and we can lose some of the exons, but we'll see what happens in the question. So immediately following transcription and RNA splicing, okay, so we've spliced, the product would most closely resemble, okay, we've got mRNA and tRNA. Well, it's not going to be tRNA, right? Messenger RNA is what's being made in the nucleus to transfer the code to the ribosome, right? tRNA is the thing that transfers amino acids to the ribosome during protein synthesis. So we're going to get rid of tRNA. That's wrong and that's wrong. So we've got a choice between choosing the exons or the introns. Now, if you can't remember which is which, exon is where you are expressing your gene. So the exons carry the code. The introns are just junk DNA. They don't do anything. Although never say never in biology. There might be an intron that occasionally turns into a bit of an exon. I don't know. Anyway, your bottom line is, as far as we're concerned, for QCAA biology, the exons are making the genes, so it has to be this one, the dark grey ones. That's the exons. It's going to be A. All right, so that's wrong. Okay. What is the most likely outcome of a homeobox Hox gene mutation? All right, the homeobox genes, they are the ones that cause repetition. So it's a lot of positional information in the embryo, and if you've got your fly embryo like this, and you've got HOX1 causes antennae to appear, right, in your fly. If HOX1 expresses over here, then that's what happens. You get antennae appearing here, because what happens is it's the pattern of Hox genes across the body that cause the different body segments to know what they're supposed to develop into. Know being a relative term in this case. They're not conscious or anything. But anyway, let's see. Slower growth rate? No. Nothing to do with homeobox. Slower growth rate would be growth hormone or something like that. Body appendages in the incorrect location. There you go. I put antennae in. They're body appendages. They will work. Failure of sex characteristics to fully develop? No, that would be some form of interruption to sexual maturation. And the impaired ability of red blood cells to carry oxygen. That sounds like sickle cell anemia to me. So what we're going to do here is we're going to go B, body appendages. That's the correct answer. There's my little diagram. Okay, next question. Done those. Getting close to the end. In fact, we are at the end, are we? Question 20. Oh no, got question 18 over here. Let's have a look. I was looking at question 20. It's a wee bit tricky for me because I learned that stuff a different way. But anyway, here we go. Calcium chloride. Calcium chloride. Ooh, this is big for a multiple choice. Okay, calcium chloride, CaCl2. Chemical use in bacterial transformation experiment was conducted to determine how the concentration of calcium chloride affects the transformation efficiency of a large and small plasmids. Okay, error bars show the standard error, right? Very, very scientific of them to put that in. That's good. Here we go. Let's look at the graph. Always try and understand your graph first. That's your first thing. So the solid line is the small plasmids. The dashed line is the large plasmids. Okay. Not saying what size these plasmids are, but small versus large. Okay. So on the y-axis, we have the transformation efficiency, and this is 10 to the 4. So what's that? 10,000 colony forming units per microgram, right? That's a lot. But that would be 10,000, 20,000, 30,000, 40,000, 50, etc. So that's how you read that scale. And then over here, we've got the calcium chloride concentration in molar concentration. So 0.1 molar, 0.2 molar, 0.3 molar. Okay, let's see. So in between here is going to be 0.15, 0.25, 0.35, etc. Make sure you can get these scales clear in your head. The data suggests that data is plural. Where's the QCAA? Eh? The optimum concentration. Do they put an S at the end of it when they mean plural? I don't know. It's very strange. Datum is singular. The optimal concentration for transforming small plasmids is 0.15 molar. Right, let's have a look at that. So the optimal concentration for transforming small plasmids is 0.15 molar. Well, let's find 0.15 molar, and that is small plasmids. So it looks to me like we're going for A straight away, but let's check the others just in case. Transformation efficiency is highest at concentrations greater than 0.1 molar. There's 0.1 molar. Well, it's not true because transformation efficiency for large plasmids is at its highest at 0.1 molar. I mean, it's possible that and after that it tails off, so it's just not correct. Large plasmids have higher transformation efficiency than small plasmids. Well, that's not correct either, except in this case here at 0.4 molar. And then finally, there's less variation in transformation efficiency of small plasmids when the concentration is less than 0.2 molar. Here's 0.2 molar, here's the small plasmids. That looks pretty variable to me. Yeah, it's going from 23,000 to 50,000 and then back down to 30,000. So definitely not that. So we're going to go with the one we first thought of. But it really is important to read all the answers when you're doing your multiple choice test, because you might misread one. If you misread A and you think that's right and you put it in, read them all. And then if you see another one that looks right, then you've got some thinking to do. Okay, you've got to make sure you've read them both correctly. It's a good tip, that. All right, let's have a look at this. Electrophoresis. Electrophoresis separates DNA fragments based on, well, there's a number of different chemical separation methods, chromatography, all that kind of stuff, and other forms of electrophoresis. But for us in biology, we're just basically separating DNA by size, and the larger fragments are left behind as the smaller fragments move faster through the gel. So size, with smaller fragments traveling further through the gel, is going to be correct. So that's, again, if you know your technique, then you can just look for the answer. Size with larger fragments, the larger fragments do not travel further through the gel. This can happen in some cases where larger fragments are, say, more positive or more negative, and they can be pulled through the gel. But that's not the case in this for DNA electrophoresis. Charge with more positively charged fragments traveling further through the gel, and charge with more negatively charged fragments traveling further through the gel. So these don't really matter because the charge on DNA is the same. So it's always DNA, so you don't get more positively or more negatively charged fragments. They're all the same charge. Okay, and I can't remember which way they go. I'll have to look it up. Right, B is the answer for that one, and we'll go to our last question. And this one kind of got me a little bit, so let's have a look at this. The capture-recapture method and Lincoln index, n equals capital M, capital N equals capital M times little n divided by m, were used to monitor a population of platypuses over a 4-year period. Good use of the platypus plural, that's correct. Over a 4-year period. Experimental findings are shown. Okay, here we go, 2019, 2020, 2021. Here's the populations, 24 platypuses, 15 platypuses. Is that right? No, 14, 15, yeah it is. 19 platypuses and 21 platypuses. All right, let's have a look. The table shows data from 1 year of the study. Now I got a bit confused about this because QCAA seem to be using these values here. It must come from a textbook in Australia. I remember this being N and K, where I had capital N equals N K over little k, where these are the recapture things. So anyway, let's see if we can figure out what these numbers actually mean. So I'm guessing that that's the total population is n. Total population is n. I'm going to say that these are both m, so I'm going to say that these both must come from the second sample, right? And that's going to be your total number of samples captured, and the little m is going to be the total marked, and then n is your original number. It's going to change this time, so they make it a little n. I think that's right. So if that's the case, that means the number of individuals captured and marked in the first sampling, we're going to make them n, right? Little n. Going to make them little n. The number of individuals captured in the second sampling, going to make that big M, and the number of captured individuals that were marked, little m. Now, I could be wrong in this, but it won't actually make any difference to the calculation. It could be that the big M stands for the number marked in the first sampling, and the little m stands for the number marked in the second sampling, and then n is the total number captured in the second sampling. Anyway, it won't make a difference because we're just multiplying these two numbers. So that's going to give us 18 times 20 over 17. That's going to equal 18 times 20 equals, I always hit the equal sign, I'm old fashioned. We didn't have brackets in the calculators when I started learning this stuff. Divided by 17, 21.17. Now what does that come close to? That comes close to, okay, so when was the data in the table collected? So this table, which year did those data come from? Well, it's going to be the one that has a population close to 21, and I'm going to say 2022, which is D. All right, that's my answer, is D. Looks like it's correct in the sheet. But as I say, I learned it this way. I can't remember. I can't see quite how that matches up, but the same thing applies. As long as you have the definition here. If you did learn it this way, all you need to do is apply your definitions. If you wanted to do NK over K. Okay. Anyway, there you go. There's the 20 questions of the multiple choice paper done as a bit of revision for you and a bit of practice for me. So hopefully I'll see you in the next video. Enjoy the exams.